And I will call Sanne Todorov, also a PhD student, but at the research group for microbial biotechnology and biorefining. And you'll talk about fungal-based protein on industrial side streams.
Hi everyone, my name is Sanne Todorov, and as just said, I'm going to talk to you about this fungal-based protein, and we're looking at side streams. I'm not sure where to stand, but I'll try and not make too much disturbance. So a little bit of background. Probably most of you know that we're going to be a lot more people in the future, and we'll need to feed those people in the future, and the best way to do that is in a sustainable and healthy way. And to keep it sustainable, we should try to minimize the damage that we do to the earth, so we should not try and get more agricultural area. We should try and keep it to what we have currently, and we should try and not burn off the forest. So we don't want that. We also don't want to introduce more animals, because we know that animals produce a lot of CO2. They produce a lot of, some of them produce a lot of methane, and generally they are just not very good at converting the protein found in plants into protein that we can eat. So we're also not looking at using animals. And we want to keep it healthy, because we need to be able to live. We need to be able to get enough nutrients out of it. So that's the big green tick mark. And the way that we are doing that is looking at these side streams. And you might ask, what is a side stream? Well, side streams are big leftovers from the industry that could be from plant-based, it could also be from animal-based, but what I'm currently looking at is plant-based side streams. So some of the side streams that you can see up here is from rapeseed oil production. We have rapeseed cake. We have, from the brewery, we have spent grain. We have apple pulp from leftovers from the juice industry. And then we have the sugar beet pulp after the extraction of sugars. And we take these side streams, and what they're currently used for today is mainly for either feed for livestock, or they're used in the energy production. So either we burn them off and get energy that way, or we use them as a medium for cultures to produce biogas. But what I'm looking at doing is instead using bacteria and fungi to produce some healthy new alternatives to meat and dairy. And the way that I'm doing that is looking at fermentation. And fermentation is just a fancy word of putting in some material and getting something else out. So basically these guys are converting some of the sugars and starches that we cannot use and producing something that we can utilize. And one of the projects that I'm working on is to use oyster mushrooms for breaking down the starches that we find in sugar beet pulp. So I'm doing pre-fermentation with lactic acid bacteria, and lactic acid bacteria is the type of bacteria that you find in yogurt or in some acidic drinks as well, sourdough as well. And we pre-ferment this sugar beet pulp that we get from the industry, and then we add the mushrooms or the mycelia to this side stream, and it breaks down all of the starches, all the cellulose and hemicellulose that we cannot utilize, and we get out this fungal mycelium, which is edible, and it's easily digestible, and it's been based on basically waste. So we get new food out. And here I'm showing a little bit older version of our project, because we actually have tried to do some prototypes, and we don't even need to dry this mycelium afterwards, because it have this meatball-like texture. So you could actually just fry it as it is and eat it as meatballs or meatball substitutes. So we're actually skipping even a step in this project. Another one that I'm looking into is again with oyster mushroom, that I'm looking into many different side streams instead of just the sugar beet pulp, to see if we can get optimized growth, to see if it grows better, if it produces more protein, or can give a better digestibility. And this is a little more complicated, but basically I'm just analyzing what is happening in there, what does it convert, what does it produce, and what can we use it for. And the last one, that's where I'm looking at new fungal strains that are not currently used in larger scales, because oyster mushroom is like a model organism. It's the one that everyone is using. So I've been looking at which other strains can we use, and especially which one can we use together with lactic acid bacteria. Because by pre-fermenting with lactic acid bacteria, we get to preserve the side stream, so we would not need to either dry it or to put preservatives into it or anything else. We could preserve the stream until our fungi is ready to use it. And what I found in here was actually that sometimes we get these interactions between the bacteria and the fungi where we get new morphological types. So in the pictures below, you can see when there's growing fungal mycelium on a acidified media, it grows in these pellets. But when it grows together with the lactic acid bacteria, it lumps into these big lumps. And that makes it much easier to harvest than if you have to harvest them in pellets. So there are these benefits to growing them together that is not just about preserving. And on the other side, you can see another strain of fungi, also mycelium, that has changed the color depending on what you're growing it on. So it's the same media that I've grown it on, but it has been acidified with hydrochloric acid, or it has been acidified with different bacteria. And you can see there's a big change in the color. That means that we could have different type of patties made on this mycelium that has different colors without having to add anything in the end, which is pretty cool, I think. So this led us to a new project that we are currently working on. That is with one of the strains, the one that changes color, that we want to see how well does that grow in different side streams. But that's where we are at now. So thank you very much.
Thank you so much.